After watching this video, you should be able to describe gluconeogenesis, or the synthesis of new glucose, which takes two pyruvate molecules and makes a glucose. You should recall the major organs that carry out gluconeogenesis, list the new enzymes that go around the irreversible steps of a glycolysis, including the substrates and products. You should be able to list the major sources of pyruvate and how NADH, NAD+, is balanced within the cell. Recall that gluconeogenesis is an ATP consuming process, and you should be able to describe the basic hormonal regulation of insulin and glucagon during the fasting and well-fed state. Now let's start with the location of gluconeogenesis, and primarily it's the liver. It carries out most of gluconeogenesis, especially early on during a fast, and the kidney to a lesser degree, and the kidney becomes more significant as you go deeper into a fast. And there might be a tiny amount occurring in the small intestine that isn't shown here. Now, the function of gluconeogenesis is to stabilize the plasma glucose during a fast. So you're fasting, you're not eating any food, your blood sugar is starting to fall, and what prevents you from becoming hypoglycemic, especially after you've gone through your glycogen stores, is the full-blown gluconeogenic pathway. Okay? Now, the regulation, as we would expect, since we're talking about fasting and well-fed state, would involve glucagon and insulin, and we're going to go into the details of that at the end of the video. Now let's go over the basics first and look at a familiar figure where we're comparing the reactions side by side. We can see here gluconeogenesis is grouped with glycogen breakdown, fatty acid oxidation, and ketogenesis. And the opposing reaction on the left side, we have glycolysis, which is lined right up with gluconeogenesis, and that's grouped with glycogen synthesis, fatty acid biosynthesis, triglyceride synthesis, and pentose phosphate shunt. And you can see here, as we'd expect, glucagon turns on gluconeogenesis and all of its associated reactions, and at the same time turns off glycolysis. And we would expect that to be high during the fasting state, and insulin to be low during the fasting state, with the reverse profile of high insulin and low glucagon during the well-fed state. Now keep in mind that while all these things are happening, I just want to point out that fatty acid oxidation is very important because it provides ATP, which is going to help run gluconeogenesis, and we'd also see a lot of ketogenesis occurring as we go deeper into a fast. So is a very important consideration to think of all these reactions in the liver when you're going over any individual one. Now, if we go and look at a more drawn out schematic, we can see here we have our hepatocyte, and let me just focus your attention over to the left side in the fasting state. We have our GLUT2 constitutively expressed transporters on the liver, and you see here we have low glucose outside the cell, and we need to generate a high glucose concentration inside the liver cell so we can go down its concentration gradient and resupply the plasma to stabilize the plasma glucose, so places like the brain and red blood cell and other insulin-independent tissues can use glucose for energy and ATP. Now this glucose is going to come from G6P, the immediate precursor, and the G6P, one source that we discussed in a glycogen video, is glycogen breakdown or glycogenolysis, which produces G1P that's eventually converted to G6P. Now that's pretty early on in the fast, and once you run out of your glycogen stores, you really start to turn on the gluconeogenesis pathway full force, and you have your 2-pyruvate going all the way to G6-phosphate, and finally to glucose and out the cell. And I just want to point your attention again, and we'll go into this more detail when we have a lecture on fatty acids. But you can see here we have fatty acids that are coming from fatty acid release from adipose cells, taken up by the liver, oxidized. You have acetyl-CoA, which can be made into ketone bodies, which is important later on in the fast. And the whole process of fatty acid oxidation, you make ATP through production of NADH and FADH2, which go into the electron transport chain, and acetyl-CoA, which can go into the TCA cycle and make more ATP. So it's just important to point out that this fatty acid oxidation is providing the energy to drive gluconeogenesis, which is really important. Now, if you go on the flip side in the well-fed state, you have glucose entering the liver cell, same transporter. There you have your GLUT2. It's going to be phosphorylated by glucokinase, the G6-phosphate. And then in this case, you make glycogen through the glycogenesis pathway. You also have the pentose phosphate shunt, which makes NADPH, which is necessary for reductive biosynthetic reactions like fatty acid synthesis and cholesterol synthesis. And the G6-phosphate can go down the rest of glycolytic pathway to make pyruvate, which can go into the mitochondria, make acetyl-CoA, which can go into the TCA cycle, and also provide carbons to make fatty acids and cholesterol. So these reactions side by side are opposite directions, and when one of these pathways is turned on, the other one must be turned off. So during the fasting state, when we're running gluconeogenesis, glycolysis is turned off. And in the well-fed state, when we're running glycolysis, we have gluconeogenesis turned off. And that reciprocal inhibition is a very important consideration for all of our reactions because we don't want to have futile cycles. Now, if we go and look at glycolysis first, before we go into gluconeogenesis, we can see that that would look something like this. We have glucose going into the liver cell through GLUT2, and you can see that ultimately we end up with 2-pyruvate, and we have these enzyme steps in red highlighting that they're the irreversible steps. And what that means is that we cannot go backwards from glucose 6-phosphate to glucose using glucokinase in reverse. We can't use PFK1 in reverse, and we can't use pyruvate kinase in reverse. So that means that we're going to need new enzymes that are special and different for gluconeogenesis to get around these thermodynamic roadblocks. Now, all the other reactions that are reversible here, they're all going to be used by gluconeogenesis because they can just simply go the other direction. Okay? So, an important point, we're going to have new enzymes that get us around these red enzymes here. Also, I just want to point out that the glyceraldehyde 3-phosphate dehydrogenase step, which normally when you're going in glycolysis makes NADH, remember we discussed that we need to regenerate the NAD plus to keep glycolysis going. When we go in the reverse direction and we're using NADH to make NAD plus, we need to regenerate NADH. So we'll need to discuss how that happens as well, just like we did for glycolysis. Okay? So now we can go into the actual pathway. Looks something like this. And here's our liver cell. And notice that what we have as being different is that we still have the GLUT2 transporter, but now glucose is going out of the cell, right? Because we're in the fasting state when we're considering gluconeogenesis. And notice that we have in red the new enzymes that are going to get us around those roadblocks. Now, these aren't drawn with the proper location inside the cell. It turns out that pyruvate carboxylase, this first step, is actually in the mitochondria, and PEP carboxykinase is probably found 50-50 in the mitochondria and in the cytosol. And the glucose 6-phosphatase enzyme, this last step, is found in the endoplasmic reticulum. But for this diagram, we're just drawing them just schematically showing the direction of things. So if we start with our 2-pyruvate, we need to carboxylate it, okay, with pyruvate carboxylate and make oxaloacetate, and that takes ATP. And the reason why we have 2 ATP shown here is that we have 2 pyruvate molecules, okay. We end up with a couple of oxaloacetates, and then the oxaloacetate can undergo a carboxykinase reaction by PEPCK, where you lose the CO2 that was added, and then you also need a couple of ATP equivalents, in this case GTP, okay, to make the phosphoenolpyruvate. So you can see that we needed 2 enzymes to get around the roadblock of pyruvate kinase, which is the last step of glycolysis, which in this case would be turned off. Now PEP can go backwards, and it continues to go backwards. Even the PGK, the kinase enzyme step that's part of glycolysis, when it goes in reverse, uses a couple of ATP. So now you see that this is where the ATP consumption comes from. We have 6 ATP equivalents to take 2-pyruvate all the way to glucose. And you can see that the pyruvate carboxylase step, the PEP carboxykinase step, which are these are the new enzymes of gluconeogenesis, and also the PGK in reverse, that's the step in glycolysis going backwards, those were all the ATP consumption is occurring. Now, going through the reverse of glyceraldehyde 3-phosphate dehydrogenase, that's where we take our NADH and go to NAD+, and we're going to need to regenerate that NADH to keep this step going so we can keep gluconeogenesis going. Now, once we have the glyceraldehyde 3-phosphate, it goes back to F16 bisphosphate, and now we encounter another roadblock because PFK1, the third step of glycolysis that's irreversible, we can't go backwards in that enzyme. That enzyme is actually going to be turned off, and we need a new enzyme, and that's going to be fructose bisphosphatase 1. That gets us to F6 phosphate. We go the reverse of a glycolytic enzyme here to G6 phosphate, and then we need another enzyme to get around that first step of glycolysis that's irreversible. It would be glucokinase or hexokinase, and that enzyme is glucose-6 phosphatase. All right? So our four enzymes that are new and different are pyruvate carboxylase and PepCK, which gets us around pyruvate kinase. We have fructose bisphosphatase 1, which gets us around PFK1, and we have glucose-6 phosphatase, which gets us around the glucokinase or hexokinase step. Okay, and notice that we are consuming ATP, a total of 6 ATP equivalents from 2-pyruvate to glucose, and we are oxidizing NADH to NAD+, and we're going to have to regenerate that, which we'll discuss in a second. Keep in mind again that since this is an ATP consuming process, that ATP is going to come from fatty acid oxidation, which is going to generate lots of ATP to keep this process going. Now, what we want to go into more detail is, well, where do these pyruvates come from? How do we regenerate the NADH? And go into some of the details of the compartmentalization of some of these enzyme steps. So that will be shown here. Okay, and we can see that what we have are lactate and a gluconeogenic amino acid or glucogenic amino acid, alanine. There's a bunch of other amino acids that can be converted to glucose ultimately in the liver, but we're going to use alanine as our prime example. Okay, and there's transporters in the liver to take these guys in. Okay, and once they're in, they can be both converted to pyruvate, but through different enzyme steps. You can see here for lactate going to pyruvate, this is simply the reverse of what we saw when we were doing anaerobic glycolysis. In this case, now we're taking lactate and going back to pyruvate through the lactate dehydrogenase enzyme step, which now is going to take NAD+ and now regenerate some of that NADH that we wanted to regenerate for the glyceraldehyde 3-phosphate dehydrogenase step. Okay, so that's one way we can do it. Alanine, if that comes into the liver, can be transaminated through an alanine transaminase, ALT, to pyruvate. That just interconverts to alpha-ketoglutarate and glutamate. Recall that that's going to be important integration of gluconeogenesis in the urea cycle, which we'll discuss in another video. But when you're driving alanine to pyruvate here, you're also going to be turning on urea cycle as well, which isn't shown in this cartoon. Now, once we have our pyruvate, it can get transported in the mitochondria, and now once the pyruvate's in here, it needs to go to oxaloacetate, okay, and in this case, it's the pyruvate carboxylase enzyme, that's a mitochondrially located enzyme, is going to do that. And the alternative pathway that pyruvate could go, that we would normally see in the well-fed state where we have lots of energy, is the pyruvate dehydrogenase enzyme, which makes acetyl-CoA. That's actually turned off. And it should make sense why that would be, right? We don't want this carbon from pyruvate. We don't want any of it going anywhere other than oxaloacetate, which will then eventually go to gluconeogenesis and glucose. We don't want any of it to be siphoned off to make acetyl-CoA, and we don't need any acetyl-CoA. We have lots of acetyl-CoA because fatty acids coming from the fat cells are coming into the liver mitochondria, being oxidized, and you're making lots of acetyl-CoA, which are either going to go into ketones or TCA cycle and eventually electron transport chain to make all that ATP so we can drive gluconeogenesis. So it turns out that PDH enzyme is suppressed indirectly through acetyl-CoA building up inside the mitochondria. And that makes sense because if you have lots of acetyl-CoA, you wouldn't want to be making more of it. Okay, so that helps by turning off this enzyme that helps push the pyruvate into the pathway we want, which is gluconeogenesis. Now it turns out that oxaloacetate doesn't have any transporters on the mitochondria to get it out of the mitochondria. So we have to convert it to some other things that could be shuttled out. And one option is it could be converted to aspartate through an aspartate aminotransferase. The aspartate comes out down its concentration gradient, and then it just goes the reverse of that aspartate aminotransferase back to oxaloacetate. So it's really simply serving as a shuttle mechanism. And you can see during this aminotransferase or transaminase reaction, just like we saw with ALT, there isn't any interconversion of NADH and NAD+. Okay? It's not an oxidative reduction reaction. Now, we also can have oxaloacetate be converted to malate through the malate dehydrogenase enzyme, and that does take an NADH to NAD+ in the mitochondria, but that's okay because when the malate comes out, it just reconverted back to oxaloacetate, and now we regenerate the NADH, which is another source of regeneration of NADH to keep that glyceraldehyde 3-phosphate dehydrogenase step going. Now, once we have the oxaloacetate in the cytosol, PepCK can then convert it to 2 PEPs, and that eventually can go back to a single glucose molecule. Now, keep in mind the way I have this drawn here, I'm showing the PepCK in the cytosol. It turns out that in humans, there is some PepCK in the mitochondria, and if that was the case, the PEP that was generated in the mitochondria would also just be transported out. But we don't have that shown in this picture. We're just showing it here in the cytosol. Okay, I just wanted to point that out. Now, I do want to mention an important point about lactate here. Lactate, which is the end product of glycolysis, anaerobic glycolysis, circulates in the blood at a pretty low level, and it's cleared by the liver. You can see that what maintains that clearance is this LDH enzyme going in reverse in the liver. That means that if there's any problem with this enzyme or NADH balance in the cell, or a problem with gluconeogenesis in the liver, lactate clearance is going to be impaired. Now, the reason why this is important is that if somebody has an underlying overproduction of lactate, like let's say they have heart failure, right? And so their tissues aren't getting blood flow, there's low oxygen, and so they're doing anaerobic glycolysis and they have extra lactate being produced, or they have lung disease or something like that. If they have those sorts of problems where they're making extra lactate, and then on top of that, there's a problem with the liver clearing lactate, you can end up with something called lactic acidosis. And so that's an important consideration because there's a couple important examples. When you consume a lot of alcohol dehydrogenase actually produces a lot of NADH and steals this NAD+ and impairs this conversion step, and that makes it a little bit more difficult for the liver to clear lactate. So if someone's drinking a lot of alcohol and then they have on top of it some other underlying problems, that increases their chances of getting some increased lactic acid in the blood, or lactic acidosis. Also, any drugs or disorders that suppress gluconeogenesis also would back things up and impair lactate clearance, and that's going to be particularly a problem if they have increased lactate production. So individuals that have mutations in some of these gluconeogenic enzymes that we discussed, for example, glucose 6-phosphatase, which is the last step of gluconeogenesis, that's actually called von Gierke's disease. It's another one of those glycogen storage disorders. In fact, I think it's type 1. Those individuals are prone to lactic acidosis. And maybe something that might come up more commonly is individuals who take the drug metformin, which suppresses hepatic glucose production, are prone to lactic acidosis if they have also an underlying condition that overproduces lactate, like we discussed, like heart failure, or lung disease, or something like that. And so that explains why lactic acidosis occurs when you have problems with this gluconeogenic pathway or NADH balance in the liver. So that's an important medical consideration for this whole process here. Okay? Now, the other important consideration is that if we go back to our picture here, notice that hepatic glucose production is supposed to be occurring during fasting. Okay, and normally when we wake up after an overnight fast, if we're sleeping, we're not hypoglycemic because we have this liver production of glucose stabilizing the glucose. Just keep in mind that if you have overproduction of hepatic glucose production, gluconeogenesis is turned on too much, that can cause fasting hyperglycemia. And that would be occurring in conditions like diabetes mellitus, where you have an imbalance of the hormonal profile, you have decreased insulin action and increased glucagon activity, and you have this process overdoing it, and the individuals, when they wake up after they're sleeping and they haven't eaten, their blood sugar's too high. Okay, and that's called fasting hyperglycemia. And you would treat that with drugs like metformin that suppresses gluconeogenesis. Okay, and we'll discuss that in another video. But I just want to point out that when the liver is not doing its job, you get fasting hypoglycemia, and when the liver is overdoing it, you get fasting hyperglycemia. Okay, now let's take a look at the regulation, which is the last consideration, and you can see that the major mechanism is probably glucagon binding to its receptors on the liver, the GS receptors increasing CAMP, eventually increasing cyclic AMP and pKa, pK phosphorylating cyclic AMP response element binding protein, and then that inducing the gluconeogenic enzymes. Okay, so that's probably the major mechanism. So you're going to get more glucose 6-phosphatase, more fructose bisphosphatase 1, more PepCK, more pyruvate carboxylase. Okay, and there's some other regulation too that occurs that we're not going to show here. Now, insulin might suppress gluconeogenic enzymes too, and so if insulin is low, that also might help get more of these enzymes going. Now, the other consideration is the fructose 2,6-bisphosphate, which we discussed in the glycolysis video. Remember that that's not only a regulator of PFK1, but it's also an inhibitor of fructose bisphosphatase 1. And so when pKa phosphorylates that bifunctional enzyme, PFK2 fructose bisphosphatase 2, and you get less of this allosteric regulator, not only are you turning off PFK1, but you're also turning on fructose bisphosphatase 1. And I encourage you to go to the glycolysis regulation video to see that in more depth. So you can see here that this summarizes when you're fasting. Here's the glucagon, pKa, phosphorylation of Krebs, induction of our gluconeogenic enzymes. And in the well-fed state, remember we have less glucagon, less pKa, less phosphorylation of Krebs, and less induction of these enzymes. And so there's other mechanisms too that we're not going to go into, like, for example, acetyl-CoA and NADH have some effects as well. But the major hormonal regulation is going to be this glucagon induction of these enzymes. And that concludes this lecture on gluconeogenesis.